Welcome to ClassMate. I'll help you learn cell biology. We begin our journey by zooming in from the human body to the microscopic world of the cell. The cell is the fundamental unit of life, a bustling factory of complex functions. Throughout this course, we'll map the interconnected processes that drive every living thing. It all starts at the plasma membrane, the selective barrier that defines the cell's boundary. This bilayer is a fluid mosaic composed of phospholipids, proteins, and cholesterol. Membrane proteins facilitate the passive movement of molecules like glucose across the barrier. Active transport uses cellular energy, or ATP, to pump ions against their gradient. Cells communicate through receptors, transmitting external signals into the intracellular space. This triggers a rapid signal amplification, allowing the cell to respond to its environment. Failures in membrane transport can lead to serious conditions, such as cystic fibrosis. Inside, a vast network of membranes, the endomembrane system, manages protein production. Ribosomes dock onto the rough ER, translating genetic code into nascent polypeptide chains. Chaperones ensure proteins fold into their functional three-dimensional shapes. Vesicles then transport these proteins along microtubule tracks to the Golgi apparatus. The Golgi sorts and modifies proteins, preparing them for their final destinations. Lysosomes act as the cell's recycling center, breaking down waste with acidic enzymes. Mitochondria are the powerhouses, generating the ATP needed for cellular work. Peroxisomes maintain balance by detoxifying the cell and neutralizing reactive oxygen. Now we enter the nucleus, the control center where genetic information is stored. DNA replication is a rapid, precise process that ensures genetic continuity. Transcription converts genetic code into mRNA, the blueprint for protein synthesis. Splicing removes non-coding regions, refining the mRNA for export to the cytoplasm. In the cytoplasm, ribosomes read the mRNA to assemble amino acids into proteins. Even a single-point mutation can lead to disorders like sickle cell anemia. As the cell prepares to divide, chromatin condenses into visible chromosomes. The cell cycle is tightly regulated by checkpoints and cyclin-dependent kinases. Microtubules form the mitotic spindle, preparing the chromosomes for alignment. Anaphase ensures that each daughter cell receives an identical set of chromosomes. Cytokinesis completes the division, creating two genetically identical cells. When regulation fails, uncontrolled division can lead to tumor growth and cancer. Stem cells are the foundation, capable of specializing into any cell type in the body. Epigenetic markers turn genes on and off, driving the process of differentiation. Specialization allows cells to perform vital functions, like the rhythmic beat of a heart cell. Cells also build the extracellular matrix, the scaffolding that supports tissue organization. Junctions anchor cells together, creating functional tissues and barriers. From these foundations, complex tissues like muscle and nerve are formed. Understanding these mechanisms is the key to the future of molecular medicine. By understanding the cell, we understand the very essence of life itself. Thank you for choosing ClassMate. See you in the next lesson.